Hi, my name is AJ from We Learn to Share, and today we'll be learning.
Today I'll be sharing my knowledge on Unit 6, gene expression and regulation. I divided this unit into two parts. This is the first part, the central dogma of molecular biology. And in a minute, we're gonna get to know what central dogma of molecular biology is. All right, so let's get started. Here is the table of contents. Number 1, molecular structure. Number 2, replication. Number 3, central dogma. Number 4, transcription. Number 5, RNA processing. Number 6, translation. We're gonna learn these processes one by one. Number 1, molecular structure. So we're gonna talk about what DNA is. As you guys may already know, DNA, people view them as the genetic material, right? DNA is deoxyribonucleic acid. That's why they're called DNA. It stands for deoxyribonucleic acids. And it's the genetic material. And actually, people don't distinguish the genetic material and DNA. They just use them both to describe the same genetic materials, right? But the thing is, DNA and RNA are part of nucleic acids, and nucleic acids is what the genetic material is. So DNA and RNA are both the genetic materials, and these are both nucleic acids. And unit of nucleic acids is nucleotides. And as you can see in this diagram right on the right, this one unit, that's a nucleotide. And as one single unit of protein is an amino acid, so the single unit of the nucleic acid is a nucleotide. Nucleotide consists of phosphate, sugar, and then the base pairs, bases. And it's always the nucleotides, they form a bond. These bases form a bond, and they're called base pairs. And it's always A pairing up with T, and G pairing up with a C. And A is adenine, T is thymine, G is guanine, and C is cytosine. So whenever you see an paired DNA in one side of the base is an A, as an adenine, you should expect to find the thymine in the other part, the other end of the DNA strand. And as I mentioned before, there is something called RNA too. And DNA is double-stranded, and RNA is a single-stranded genetic material. So, and another difference is that RNA has a U instead of a T. It's something called uracil, which is a pyrimidine. Pyrimidine, we will explain this in the next slide. It's a pyrimidine replacement of thymine in DNA. So instead of T, they have U, uracil. So as I mentioned before, purines versus pyrimidines, what are they? Purines are those bases with a let's say these rings, two rings melded together, and pyrimidines are the single ringed bases. So there's another thing that you need to remember, and keeps popping up whenever you study genes and DNA and genetic replication, is 5' to 3'. It always says genetic material is transcribed, or genetic material is replicated from 5' direction to 3' direction. But what is 5' and 3' talking about? So starting from the base, so this is a nucleotide. On the left side, it's an RNA nucleotide. On the right side is the DNA nucleotide. And take a look at this nucleotide. There's a phosphate and there's a sugar, pentose sugar. So there's 5-carbon sugar, the pentagon-shaped sugar, and then there's a base. And starting from the carbon that connects the base, we count them as 1. And counting them clockwise, we count up to 5. And the one above here is the oxygen. Number 1, number 2, number 3, number 4, number 5. And let's go back a couple of slides. As you can see, this DNA strands are connected from starting from the phosphate here, and then 5' carbon here, and then 3' carbon connects to the next phosphate group. So it keeps on connecting 5' to 3', and then 5' to 3'. That's why it's called everything is replicated. They say that everything is replicated from 5' to 3'. So take note of that. Also, one important thing about DNA is its structure. DNA is said to be a double-stranded helix. So a double-stranded helix is basically it's a two-stranded, two-twisted strand. So as you can see, that there are two strands, and then it's twisted as a helix structure. So why does it form two strands? It's for stability, basically. As I've mentioned before, A pairs up with a T and G pairs up with a C, so they're always complementary to be stable. So there could be a single-stranded DNA, but eventually and essentially DNA is double-stranded, and they have to form a pair and complement. And as seen in this diagram, and also the diagram that I showed a couple of slides back right here, the shape is like an upside down. Although it forms a pair, the nucleotide, take a look at the nucleotide, individual nucleotides, they're all upside down. So 5' is over here and 3' is over here for this strand here. So that's why it said to be anti-parallel. And the structure was initially it started to be scientists started to suspect that the structure is double-stranded helix, starting from this picture right here. It's an X-ray diffraction image taken by Rosalind Franklin. And deducing from this, and from the fact that Erwin Chargaff found that, and what he found was that adenine and thymine, how much adenine and thymine is in a single DNA is very similar. It's equal, and the component, how much G and how much C takes up in the whole DNA is equal. So A, T, and GC is equal. That's what he found. And taking from this Rosalind Franklin's picture and the fact that Erwin Chargaff found, Watson and Crick devised this double-stranded helix structure, and which we know as structured DNA in this day. So replication. How is DNA replicated? So initially, scientists begin to think about how DNA is replicated. And from an early stage, there were three main theories, three early models of DNA replication. And as you can see in this diagram, there are number 1, conservative, number 2, semi-conservative, number 3, dispersive. And as the name suggests, and also in this diagram shown here, you could probably guess what each of the theories mean, but let me explain them for you. So conservative, you have the initial double-stranded strands, right? And then the next phase, you have the same double-stranded and the entirely new double-strand formed. So, and this next theory says from this initial double-strand, each double-strand is separated, and then the new strand is formed based off of that initial strand. And dispersive is, as you can see, it's just dispersed. Everything is dispersed into new. And so if I were to jump to conclusion, so the theory that we believe to be the actual model of DNA replication is semi-conservative. And Messelson and Stahl found this out by conducting an experiment by using isotopes and density gradient, which I will not talk about deeply in this video, but if you're interested, you can search Messelson and Stahl experiment. It's very famous, so there could be numerous resources about it. So yeah, check that out if you're more interested. And one thing you need to take away from this slide here is that model of DNA replication is semi-conservative, as you can see right here. One strand, the initial strand, is used as a template to create a new complementary strand. Next up, I'll talk about the process of DNA replication. Firstly, DNA unwinds this origin of replication, right? It starts unwinding, separating into two strands. The helicase, this one, it unzips it. Think of it as like a ziplock. It unzips it. It opens up the DNA forming replication forks. And topoisomerase here, it relieves additional cooling. Notice how some slides back, these two strands they're twisted, right? And if we were to open these up, that would create a supercoiling. So the point of these topoisomerase is to unwind those supercoiling to make it more stable. And prime is synthesizing RNA primers complementary to the DNA strand. And DNA polymerase starts adding nucleotides to the 3' end of the primer. As I said, from the 5' end, 3', new ones are added to the 3'. So the DNA replication is always 5' to 3'. Elongation of both the lagging and the leading strand continues. I will explain this in a second about the lagging and leading strand, but just understand this for now. And the gap between the two DNA fragments is sealed by DNA ligase. So DNA ligase is the enzyme that seals the separated fragment back. And it again forms the phosphodiester bonds. Phosphodiester bond is basically just a new phosphate sticking into the 3' end of the carbon. Leading and lagging strands. So as I said, helicase opens up the DNA fork, replication fork, right? And as the DNA replication fork opens up, there's going to be two that's opened up. The starting two that's opened up, and as I said, as I also mentioned before, the replication is always, the direction of the replication is always 5' to 3'. So for the leading strand, it's just easy. It starts from 5', it just never ceases, just continues. It's never disrupted. It just continues on and on, replicates without fragmenting, right? But for the other side here, it's gonna be hard to do that because it opens up from the 3' end. So the fork opens up from the 3' end. So it's really hard, since DNA replication is always, with no exception, it's always 5' to 3'. It's gonna have to be fragmented, right? Because the direction is this side, but the fork direction of the fork opening is this direction. So it's gonna have to be like, keeps on replicating with the fragment. So these lag behind these leading strands. And these fragments found by Mr. Okazaki, I believe, called Okazaki fragments. Next, I'll talk about central dogma. What is the central dogma of molecular biology? So it's basically talking about the flow of information from DNA to RNA to basically protein. And from this process of DNA to RNA is called transcription. And from RNA to protein, that's the translation. So this is the basic theory and the fundamental central dogma. It's just flow of information from DNA to protein, right? There's also something called reverse transcription from RNA to DNA, but that wasn't the first initial central dogma that the scientists imagined as a device theory. So, and it's also how proteins are made. It's from this piece of information in DNA, an actual protein that works in your body. Next up, I'll talk about the genetic code. Genetic code is how base pairs code for proteins. From DNA to RNA, message is transcribed. As I said before, from a DNA language, take it as this. From DNA language, the message has to be transcribed into RNA language because DNA is like the actual file, and RNA is like a copy file, right? And you have to use the copy file to send out a message or send it to someone else. You can't actually give the file that you need, right? So DNA has to be copied first into our RNA, which we call that transcription. And 3 letters of RNA, they code for one amino acid. So for example, this is a diagram, key read from inside out. So let's say you have a genetic code of UCG, right? Say you have a UCG. UCG codes for SER right here. And CUG codes for leucine. And for example, another example is UGA codes for, it doesn't actually code for a specific amino acid, it codes for stop codon. Yeah, and then another example, GAU codes for aspartic acid. And I talked about the stop codon. There's also a starting codon. So there's a start and end codon. And although there are multiple end codons, UAG, UAA, UGA, there's only one start codon actually. And if you take a look in this diagram, although there's these areas where it says stop, which indicates stop codons, try to take a look and try to find where it says start. There isn't one that says start, and that's because the one AUG, methionine, actually is the start codon. All the codons have to start with AUG and bring up a methionine to start transcribing. So next up, we'll actually talk about the process of transcription in detail. Steps of transcription. 3 main steps of transcription. Number 1, initiation. Number 2, elongation. Number 3, termination. Initiation is a process in which RNA polymerase attaches to the promoter. Promoter is like the initial basis that you have to start with for the RNA polymerase to start elongating. And it starts RNA synthesis, attaching to the promoter and starting. So basically initiating. Elongation is making it longer, right? RNA strand grows from 5' to 3'. Free nucleotides attach to by the function of polymerase, and then these, as you can see right here, these free nucleotides floating around, they attach to form a pair. And termination is when RNA polymerase reaches the terminator DNA and detaches. And since RNA polymerase is an enzyme, it will be used over and over again for another step of transcription. Number 5, RNA processing, mRNA processing. So there are different types of RNA actually. And the RNA that I talked about before until now is mostly just mRNA. And mRNA, M here stands for a messenger. And messenger RNA, that's like the copy version of DNA. It's a messenger. Right? And tRNA is, there's something else called tRNA. There's also something called rRNA. And there are introns and exons in an RNA, right? So in DNA, there are actually there's information that codes for proteins, and there are some bases that do not code for a protein. And those that do not code are called introns. And those that are important, those that do code are called exons. So RNA splicing is basically taking out those introns, the unneeded introns, and concentrating the exons. And also important part of mRNA processing is capping and polyadenylation. So capping is a G prime capping. G capping at the 5' end. So in the start of the RNA strand, and at the end, they add a poly-A tail, polyadenylation. So they add the adenine. And what this does is it makes the RNA strand more stable. It's like a protection. There's also something called alternative splicing. So in the slide before, we talked about the RNA splicing that takes out the exons and then reattaching them, right? Alternative splicing is a selective removal of exons. So basically, RNA splicing, it just takes out the introns and then reattaches the exons, right? Alternative splicing is where when mRNA is processed, some of the exons are reattached and some are just taken out, and it makes different combinations. Right here, as you can see, for protein A, B, C, there are all different mRNAs. That's because some of the exons were taken out as in different type of combinations. And there are 5 exons, right? And for A, there's all 5, 1, 2, 3, 4, 5. For B, 3 is taken out, so 1, 2, 4, 5. And for C, 4 is taken out, so 1, 2, 3, 5. And all these proteins, since they have different amino acids and different structure, they'll perform different functions. Next up is translation. tRNA. As I mentioned before, these are the one of the other 2 RNA that I will talk about today. And tRNA, T here stands for transfer RNA. And these are not translated. The ones that are translated are mRNAs. These are not translated. They assist in the protein synthesis, and they have this area called anticodon loop that has anticodons. So before, when we talked about genetic code, we had the codons, UAU, UAC, GCA. These are all 3-letter codons. An anticodon, this is like the complementary of a codon. So for a codon of GUA, the anticodon would be CAU. Since it was GUA, G pairs up with C, U pairs up with an A pairs up with a U. Next kind of RNA is the ribosomal RNA. It's also not translated. It's rRNA and ribosome is the most necessary component of translation. There are 2 subunits in the rRNA. There's a larger subunit and a smaller subunit. It's like 2 balls that are kind of smushed together. There's a larger part and there's a smaller part. And each of them is made up of rRNA. And there's also something called E site, P site, and A site that we will talk about in a second. And polypeptide grows out of ribosomes. So ribosome is where protein synthesis is mainly taking place. So polypeptide is grown out of ribosomes. So bigger, large subunit, and then smaller subunit. And then these make up one rRNA and ribosome structure. So steps of translation is similar to the step of transcription. And number 1, initiation. Number 2, elongation. Number 3, termination. mRNA binds to a small ribosomal subunit. tRNA binds with the start codon. and large ribosomal subunit binds. In an elongation, tRNA enters the A site with the amino acid. And then, so E site, P site, and A site is like the site inside the ribosome. We'll provide you with a diagram in a second. A site with the amino acid elongates the peptide bond. tRNA leaves after attaching the amino acid. Termination, stop codon reaches the ribosome's A site. So take a look at this diagram. There's an E site, P site, A site, and these tRNA they bring up a amino acid with them, and those that have amino acid they attach to this codon with the complementary anticodon, and then it attaches and then recognizes, and then these two form a peptide bond, and then they move to the next site, and then E site is basically exit site. So this one with an empty, the one tRNA that's not carrying any amino acid it just exits, and then it's repeated and it keeps elongating. Thanks for listening today, and if you have any questions, you can contact me in the email below, or you can visit our website and we learn to share. Thank you. See you in next lesson.